Okay, so the chromosome, it is going to consist of the DNA molecule, and it gets packed together with the protein. So remember, your chromosome is your condensed version. Let me need this a little bit bigger. It's your condensed version of the chromatin. That's where you have everything all coiled up in there. So the nucleoid is that dense region of DNA in the bacteria because they don't have the nucleus. That chromatin is the DNA and protein fit together into the nucleus of the eukaryotic cells. It's chromatin when it's not in the dividing state. That's when it's all loosely coiled. Your histones, these are going to be small proteins that over a fifth of them are positively charged with lysine or arginine to bind tightly to the negative strand of DNA. So by them having a positive charge, the negative charge on the DNA is going to want to stick to that in there. And it's going to be your first level of packaging the DNA in there. There are multiple different histones out there. With the four most common types are the H2A, H2B, H3, and H4. So those histones are going to have the DNA wrap around them and make up nucleosomes, which are beads of DNA consisting of the DNA that's wound twice around the two molecules of each of the four main histone types in there. So the nucleosome is made up of DNA wound around a histone in there. Histamines. Yeah. So the linker DNA is going to be the strings of DNA in between the beads. And in the cell cycle, the histones are going to leave the DNA only briefly during the DNA replication. So otherwise, it stays on there pretty much the whole time. So we make these 30 nanometer fibers, which are going to be your histone tails of one nucleosome, the linker DNA, and the H1 histone to interact and coil them up into a 30 nanometer chromatin fiber. So you've got these fibers of chromatin that are 30 nanometers in diameter, is what that's saying. And you're going to have loops of the 30 nanometer fiber attached to the chromosomes and be scaffolded, where it's going to have other proteins in there. So it gets wound up again for another level of organization to make a 300 nanometer fiber. So the metaphase chromosome is going to have the loop domains of the coil and folds, and then it further gets compacted into all of the chromosome into chromosomes. So with even more packing in there, that you end up with a width of 700 nanometers. And despite all of this folding that goes on to make a chromosome, you always end up with the same genes in the same place. It folds up exactly the same every single time in there. The folding is not fully understood in there, but it's pretty amazing that you take something that would be like saying you're going to take a piece of thread and you're going to wind it around and fold it enough to make a rope that's this big. And every time you do it, it's going to be exactly the same in there. But that's essentially what's happening. So metaphase chromosome is when it's made into those full chromosomes that you typically picture. The heterochromatin is the interphase chromatin. That's going to be the little clumps visible with a light microscope. This is going to be your interphase chromatin during that part of the cell cycle in there. And then your euchromatin is less compact, more dispersed. This is when it's totally loose in there. The heterochromatin, a lot of the cell's enzymes can't access it. Euchromatin, that's when it's most useful. The DNA can be accessed by the enzymes in there, and then you can actually transcribe the genes. The other two phases is more of packaging phases in there. So we should have, I thought there was a picture of that in here. I guess not. Oh, I guess we were right at the end of that section. So now we're going to look at how we go from the genes to the gene expression in there. Your genes are going to specify the protein that is made. So now we're going to look at how we go from the genes to the gene expression in there. Your genes are going to specify the protein that is made through transcription and translation. So the reason they found this, there was evidence from the study of metabolic defects where the genes would dictate the phenotypes in there. So they looked at nutritional mutants in the neurospora, and for a long time we had this one gene, one enzyme hypothesis that one gene only coded for one thing, and it was that simple and straightforward. And it is not that straightforward, we've learned anymore. In 1958, we discovered that the genes acted by regulating definite chemical events in there. So with the products of gene expression, they decided they needed to modify that hypothesis. And so they revised it to the one gene, one polypeptide hypothesis, which is still not entirely correct. That is different because you can have a protein be made from multiple polypeptides coming together to make the protein. So they looked at that and said, all right, we can have multiple genes be involved in something because of having different polypeptides in there. So a lot of eukaryotic genes are actually going to code for a set of closely related polypeptides via alternate splicing, which we're going to talk about that in more detail later on. But you've got your parts of your gene in a eukaryote, and they're broken up into these little sections, and some of the sections we don't actually express in there. And so you can have one gene with multiple sections in there, and to make different proteins, you use different combinations of those sections. And so quite a few of the genes code for RNA molecules that have important functions in the cell. And one of the things they've also recently learned is you just may go as far as making the RNA and never actually go on to the next step of making the protein in there, that you just make RNA molecules that you'll use to regulate other things. So when we look at the basic principles of transcription and translation, the DNA provides your instructions for making proteins, but it does it through an RNA intermediate. So in a eukaryotic cell, you have this DNA, and you need to send that set of instructions out to the cytoplasm. But the DNA won't fit through the pores in the nucleus to get out to the cytoplasm, so we make an RNA intermediate in there. So transcription is the synthesis of the RNA using the DNA as a template. So where we have those A and T's that pair together, the G and C's that pair together, when we copy to make another piece of DNA, you do a very similar process in there to make an RNA, and then that RNA gets sent out into the cytoplasm in there. So what this picture shows right here, particularly this one, if you understand it in a eukaryote, it's even simpler in a prokaryote. You've got your double-stranded DNA, you use it to make an mRNA, and then that mRNA goes out into the cytoplasm where you translate the code on it. We call this part in the nucleus transcription, where you're writing a copy of it, then where you're actually reading it, that is going to be your translation in there. So the mRNA is your messenger RNA, is where the M comes from. And that's going to have the gene's protein building instructions, and it's going to carry that message from the DNA to the host protein synthesizing machinery that's out in the cytoplasm. So do you remember from the cell which organelle we associate with protein synthesis? Starts with an R. The ribosomes. And so that's who it's going to send it to. So translation is going to be the synthesis of the polypeptide that's going to use the information in the mRNA. It's going to translate that coded set of instructions in there. And then the ribosomes are going to be your organelle. They're the complex particle that facilitates the orderly linking of amino acids in there to make the polypeptide chain. That's a very formal definition for saying it's going to read the code along the way. So in a eukaryotic cell, your transcription is going to occur in the nucleus and translation in the cytoplasm. In prokaryotes, you don't have a nucleus. So you're going to have it all occur in one compartment in there. In a eukaryote, you have to have transcription be finished before translation starts because they're in two separate compartments. You have to finish the one to send it out to the ribosomes. However, in a prokaryote, they can actually start to read the code before they get the full set of instructions in there. So your eukaryotic RNAs are going to also be modified in ways prior to leaving the nucleus before it's going to make your final functional RNA. So in the prokaryote, they just take the instructions the way it is. In the eukaryotes, we want to clean up the instructions before we send them out there. So your initial uncleaned up RNA is considered your pre-mRNA. And it's also sometimes referred to as the primary transcript in there. So your genetic code, we only have 4 different nucleotides in there, and we have 20 different amino acids. So that means we can't code one base pair for one amino acid. There's not enough. We can't do a combination of 2 because that's only 4 times 4 possibilities. Still not enough. So what we have to do is combinations of 3 in there, which is going to give you 4 times 4 times 4, 64 possible combinations in there. So that also means you're going to have some duplication in that code in there. And we refer to it using a triple code in there, or we call them base triplets. They get a few different names in there. But it's in a series of 3 nucleotides that are going to code for each amino acid that's used. So here it's showing, if we take just a small part of your chromosome here, where we've got a section here that's a gene, and this is the DNA section of it. You've got your G and C's paired together, your A and T's paired together. When we make an RNA, remember, is a little bit different. It's going to use uracil instead of thymine. So you're not going to see T's in this anymore. You're going to see A and U's paired together. That's one of the things you can look at that gives you that clue that you're dealing with RNA instead of DNA. So it's going to take these in 3s and read the code that's going to tell you what the amino acid is. So each time you come to a new 3 of them, a new set of 3, a new set of 3, it will tell you which amino acid it is. And there's actually a table that you can use that will tell you, and you can look at that and find out specifically which amino acid is going to be coded for by which 3 base pairs in there. So that's not something you would memorize. No one would memorize it. Generally, in a lab, you would never sit down and sequence it anymore. It's all computerized. So your template strand is going to be the strand of DNA that you're going to use as a template or your pattern for making the RNA. So just like when you make the complementary DNA, when you're copying your DNA like we talked about in the last couple days, you're going to use that template to make the complementary RNA that's going to fit on it. So instead of filling it in and using thymines to pair up with the A's, you use uracil. So for any given gene, the same strand is used as the template every time. So when you look at the 2 strands of DNA here, which one is the template and which one is the complementary strand? It varies with the gene. So you may have gene A be right here, B be right here, and gene C and D. Every time you use it, gene C is always going to be on this side. But it is random on which side of the strand is considered the template that we use. So the RNA pairs similar to DNA substitutes uracil for thymine, and then it's going to be synthesized in anti-parallel directions to the template strand. So it's going to be synthesized 5' to 3' end, just like DNA is. So your codons are going to be those mRNA triplets that are going to code for the specific amino acid. They're generally written in the 5' to 3' direction in there. Since we read DNA 5' to 3', it's just a convention that we generally write everything out in the 5' to 3' direction to keep it consistent. So of those codons, 61 out of the 64 of them are going to code for amino acids. 3 of them are going to code for stop codons. They don't actually code for anything, and that's why we call them a stop. And that's going to mark the end of translation. So when you get to a stop, that's how you know to stop reading in there. We know where to start reading because it always starts reading at the first AUG. And AUG is going to code for methionine in there. So that one you usually end up remembering because it always starts at AUG and always codes for methionine. So just out of the sheer repetition of it, you remember that one. But the other ones you would always look up on a table. So in the code there is redundancy where you can have multiple codons code for the same amino acid, but there's no ambiguity. There's no one codon that's going to code for 2 different amino acids and be an either-or situation. So your reading frame is important. It's going to allow the correct message to be read depending on the symbols in there. So when you're reading it and you go along the sequence, we know our reading frame when we look at this. It's coming along looking for the first AUG is right here. So our reading frame is like this. If you were reading it in a different frame, where another color would be helpful, where we started to read in this frame, it's going to code for something entirely different. So that reading frame is important, and finding that first AUG is what's going to establish your reading frame. The genetic code is nearly universal, which is convenient because you can take and transplant genes from other organisms into something else. It's allowed us to be able to manipulate things because of the universal nature of it. There are a few little exceptions where there are these translation systems that are going to have a few different codons than standard ones. We haven't found anybody that uses anything that's entirely different yet in there. And that variation has been in some of the unicellular eukaryotes and in some of the organelles. I haven't been able to find a lot of information on the exceptions because for a long time we didn't think there was any exceptions. We've just found a couple that have a few little tweaks in the way they handle the rules. So rather than memorizing it, you use this little chart. And the way that you use this chart is when you're looking at the base pairs, you find the first base pair along the left. So when we look at AUG, you find A here. Your second letter in the triplet, U, you find across the top. So when you come together, bringing the A and the U together, then you look for where the third one is over here, and that will tell you which one. So there's multiple G's here, so you want to go into that square region where these are, and that will tell you what it codes for. So if you look at that, what is CCG going to code for, looking at that table? PRO, the proline in there. So you went C, C, and then a lot of times you can just look in the box when there's only 4 to choose from and find it just as quick that way for proline in there. Is there anybody that doesn't see how you go about finding that in there? So it's pretty straightforward. This is always the codon that you're looking at in there. You can find these tables that will go back and they'll have it translated back into using it from the DNA, but the easiest way to take it is looking at it as if you were reading the mRNA in there. That's where you start reading. So that tells you your frame of reference in there. You read them in threes. So with the example here, where we had the AUG and then the next one was CCG, so this gene is going to continue on. So if the next one was GCA, then you would look on there, and you just keep reading 3 at a time like that till you eventually come to a stop. And our 3 stop codons are the UAA, UAG, and UGA in there. I have seen things where they have named the stop codons. For the most part, nobody names them other than a few sources, and I've never seen the names referred to in any kind of research or literature outside of somebody just being anal naming things, I think. I think they just weren't named and somebody wanted to put their name on something. Nope, you would always have a table to work with. Yeah, I have a few of them that I have memorized because of using examples on the board, but they serve me absolutely no other purpose in life. So, transcription is the DNA-directed synthesis of RNA. So, in order to do this, this is going to work a lot like copying DNA does. You're going to have an RNA polymerase that's going to go on and do the polymerization or adding the extra pieces on instead of a DNA polymerase here. So you use the DNA polymerase when you're adding on the extra nucleotides to make DNA. You use an RNA polymerase when you're adding extra nucleotides on to make RNA. So you're going to split the 2 strands of DNA apart, and just like you did with the DNA, and it's going to join the RNA nucleotides together in the 5' to 3' direction, just like you did with the DNA. And you're going to need a primer to start. So it's going to be the DNA sequence in there that allows you to start, just like you had to have with making DNA. The promoter is going to be the DNA sequence where the RNA polymerase attaches to initiate transcription. So that's going to be a little bit different in this. The promoter is going to allow that attachment on there, and so it's also a site of regulation of, do we want to make this protein or not in the cell. And then the terminator is the sequence at the end that signals the end of transcription. There is some variation in how prokaryotes and eukaryotes are going to end the process, but you do have the stop codons in all of them. So the transcription unit is going to be the stretch of DNA that is transcribed into the RNA. So your bacteria have a single RNA polymerase that is used for all types of RNA, for ribosomal RNA, mRNA, and your tRNAs, any of them out there. Eukaryotes have at least 3 types of RNA polymerase in there. Again, we're still finding these, so at this point when we talk about it, if you know it's an RNA polymerase, that's going to be enough to get you through on things. So when we make this RNA transcript, you have your starting point in there where the nucleotide, it's going to be the nucleotide where the RNA synthesis actually begins. A lot of times your promoter, which is shown down here, is going to be a regulatory sequence that is going to be quite a bit ahead of the game. Sometimes the promoter is right next to it, but the promoter is going to be before that. So we have lots of transcription factors. These are going to be largely in the eukaryotes. There are proteins that are going to help to mediate the binding of the RNA polymerase on the for the initiation of transcription. The transcription factors are what we're going to use to help regulate this process in there. So only after the transcription factors attach to the RNA polymerase and it's going to be RNA polymerase 2 in this case. It's going to bind. Then the prokaryotic, only after these bind on there, then you're going to be able to bind and specifically recognize the promoter. So it's just RNA polymerase in the bacteria. It's RNA polymerase 2 in eukaryotes in there. All I would ask you and expect you to know is that it's an RNA polymerase enzyme in there. That's going to make your transcription initiation complex. It's going to bind to the promoter. They have found this thing that they call a TATA box in there, and it's a sequence in the promoter that contains several of these TA nucleotides in there that are upstream from the transcription for the start in there. So it's just going to be more a part of a regulatory sequence. We don't entirely understand it. That is not the same tatas that they are talking about when you see the Save the Tata signs in there, but I always think that's kind of, knowing that there's this Tata box out there, I always look at those signs and I'm kind of like, that's a little weird. So you've got your promoter here, your transcription unit. In initiation in there, you're going to have your RNA transcript, things get unwound in there, and you're going to be making the non-template strand, and then you've got your template strand. Then when you're going to be letting it come back together, right here is where you start reading the RNA in there, the RNA transcript, and it's going to come along, and then it allows it to close back up, and you make this RNA transcript there that is going to be sent back out. And when you're done, it's closed up, that completed RNA transcript goes on its way, and you've still got the DNA. And so the promoter ends up going down the stream in there. So this interaction between the transcription factors is part of the protein-to-protein interactions that help to regulate the process in there. The RNA polymerase is going to untwist the helix and expose to 10 to 20 nucleotides at a time in there. And then that's going to add your nucleotides onto the 3' end. And that RNA is going to pull away from that DNA template as it goes, and then the DNA helix will recoil. This is going to occur at a rate of about 40 nucleotides per second in eukaryotes. So it can read it fairly fast. It can read it faster in some organisms. It can go pretty fast in bacteria. So single genes can simultaneously be transcribed by several RNA polymerases. So if you want to read a gene and you want to have multiple mRNA transcripts, you can have multiple RNA polymerases hop on there, and you can make several set of directions to send out into the cell. So if you think of it being like blueprints, plans for building a house, and you and all of your relatives want to build the same exact house, and you all live in 5 different states, you can make 5 different copies of those plans and send them to 5 different states in there. That's what the cell is able to do by having more than one RNA polymerase hop on there. So the termination mechanism will differ in the eukaryotes and prokaryotes. In bacteria, the transcription needs to proceed through the terminator sequence in the DNA, and the terminator is going to be your termination sequence. In eukaryotes, they always have to make things a little bit more complicated. They will have this polyadenylation signal sequence in there, where you've got this AAU-AAA found in the pre-RNA. That's going to be 10 to 35 nucleotides downstream, and then it's going to cut it free in there. So you have this sequence that's going to be on there that is part of what it binds a little bit differently in there. It's held with not quite as strong of a bond, and so it allows it to kind of be like a tail that hangs loose and pulls off the rest of the chunk, is what they think it does. So it's kind of like if you walked around and you had a tail and it got snagged on things, it's going to slow you down, and it just snags its tail that pulls it off. So the eukaryotic cells need to modify this RNA after transcription. So after you've made this RNA, it can't be just good to go the way it is. Of course, we have to play around with it a little bit more, and we call this the RNA processing. So after your pre-RNA is modified, it has to be modified before the genetic message is dispatched. Both ends are usually altered, and a lot of times you alter stuff in the middle too. So how we alter the ends, you can have this 5' cap, which has a modified form of guanine on there that's added onto the 5' end after transcription of the first 20 to 40 nucleotides. So you can put a cap on the end. The poly A tail in there is going to be at the 3' end, and a lot of times an enzyme is going to come along and add between 50 and 250 more adenines on the end. So you're going to basically make this long tail that's going to yank it off of there and help it decide where it goes. So they're both going to help facilitate the export of the mRNA from the nucleus. It helps to protect it from being degraded by hydrolytic enzymes, and it's going to help them attach to the ribosomes at the 5' end. So you can kind of think of it being like a lizard's tail, that it can get caught and it can get chopped off, and it allows the rest of it to go on without it. That's kind of what the poly A tail is like on there. So we've got this big long gene here that's our pre-RNA. You've got this 5' cap that you've put on the end. You've got this poly A tail on the other end. And then we have these introns and the exons in here. So the introns are considered to be intervening sequences in there. They're not actually transcribed. They are just chunks of DNA in the middle. And then you've got these exons. These are the expressed sequences. So the name exon comes from being expressed in there. So when you remove the introns and you splice together the exons, now you actually have the coding segment in there. So what you actually use out of that piece that you transcribed is just a small amount of it in there. And you could have this one gene that's going to decide, you know what, I'm just going to use these first 2 exons to make this protein A. I'm going to use these 2 exons to make protein B, just these 2 to make protein C, and all of them for protein D. So that's how you can have one gene end up coding for so many different proteins. Is it still going to pick and choose which of those exons it wants to use? So they have these things they call SNRPs. They're small nucleotide ribonucleoproteins that are short nucleotide sequences at the end of each intron. And it's going to be used as a signal for the RNA splicing. So when it's deciding which of those introns and exons it wants to use, the SNRPs are going to help it in its decision making in there. Your snRNAs, your small nuclear RNAs, are going to be about 150 nucleotides long in there. So the spliceosome is going to use several of these SNRPs in there, joined with additional proteins in there. So your spliceosome is going to interact with certain sites along the introns, and then it's going to release introns to be degraded and join the exons to make that actual piece in there. So this is showing how you've got that pre-RNA strip, and these are going to help to make the decision to cut out this intron section here that you don't need. It's cut out, sent away, and ending up with your final product at the end. So ribozymes, these are RNA molecules that function as enzymes. So not all of your RNAs end up being made into proteins themselves. The RNA itself can act as an enzyme in there. In some organisms, you're going to see RNA splicing occur without proteins, and the introns actually function as ribozymes and catalyze their own exclusion in there. So to say that those introns have no function, it isn't that they have no function. We cut them out and they get used for something else. And to be enzymes, as ribozyme is one of the things they can do. So why can RNA act as an enzyme? Its single-strand nature allows it to have complementary base pairing in there. So because you have only one strand of it, and you've got A, U, G's, and C's in there, it can potentially bind with other A, U, G's, and C's on the other side. And that can help in making its three-dimensional structure, and it can help catalyze other functions in there, just like another type of enzyme would by holding things in place. Some of the bases in RNA are going to have functional groups that can participate in the catalysis, and it also has the ability to hydrogen bond with other nucleic acids in there. So it's got those properties that will allow it to do that. So why have these introns anyway? It allows a single gene to encode more than one type of polypeptide. So many genes are going to give rise to 2 or more polypeptides depending on which segments it treats as exons in there. The alternative splicing, this is where you take different combinations of which sections you use to make different proteins out of the deal. The domains are the discrete structural and functional regions in the modular structure of proteins. So exon shuffling, this is going to allow the introns increase the probability of crossing over between the exons of the allele. So you can actually have crossing over of the little sections within the gene. So you don't just get crossing over on a bigger scale of a whole gene or multiple genes moving on a chromosome. You can have crossing over of this stuff within the gene in there to give more combinations of exons and proteins. Why don't we go ahead and take a break here for a minute? So translation is considered the RNA-directed synthesis of the polypeptide. So translation is considered the RNA-directed synthesis of the polypeptide. So some of the tools used in translation here, one of them is the tRNA. tRNA stands for transfer RNA, and it's going to transfer the amino acid onto the growing polypeptide in the ribosome. And it's basically going to be this piece of RNA here that is folded up in this position, and it's got this region on it called the anticodon that is going to be complementary to the codon. So on one end you've got an amino acid hanging on there, and on another end you've got this part that is going to be complementary to the codon. So when it comes down, you can have those two complementary ends meet up with each other, and it confirms, yes, you have brought the right tRNA with the correct amino acid here, so that you make sure you get the correct amino acid added on there. The anticodon is the nucleotide triplet at the opposite end, it's going to be single-stranded RNA, and it's single-stranded RNA here. There's only 3 base pairs there, but the tRNA itself is only about 80 nucleotides long. So part of what goes into making up the tRNA, you've got an enzyme that's involved with it, the amino acyl tRNA synthase in there, that is going to make sure you get all the right things in your tRNA. So one of the things that we have found is that we only have about 45 different tRNAs. And we have the 64 different codons, 61 of them that actually code for something, 3 that are the stop. So what that suggests that there are some tRNAs that can bind to more than one codon. When you look at that table, that codon chart in there, you'll notice the thing that varies a lot is the 3rd base pair in there. A lot of times, all 4 of them in the section are going to code for the same amino acid. So if you looked at CCG, CCC probably codes for the same thing. So they use this, what they call wobble, which is the flexible base pairing in the 3rd position of the mRNA. So the rules are a little bit looser there, so it can pair with potentially CCC or CCG so that it can share a tRNA for that same spot in there. Your ribosomal RNAs, these are going to be part of your ribosomal subunits. They are going to act like an enzyme in there and helping the ribosomal RNA be able to function in the ribosome, making the actual protein in there. So the ribosome facilitates the coupling of the tRNA anticodon with the mRNA codon to make sure that they match up. You've got a large and a small subunit that are going to be involved with that. So bacteria and eukaryotic ribosomes are similar in structure and function. However, the eukaryotic ribosomes are a little bit larger and there is slight differences. Those slight differences in there are a lot of times what antibiotics are going to target. So an antibiotic is generally not going to work if you have a parasitic infection from a eukaryote, because if it's going to target a bacterial ribosome, you got to use it against a bacteria in there. It's also the reason why antibiotics are not going to work against viruses, is they don't have ribosomes to target. So a lot of times, patients will be like, well, I want an antibiotic for that. And it's viral, it's not going to do you any good. The only benefit it's helping them is psychologically, it's acting as a placebo in there. But that's actually a really good thing to have antibiotics target that, because that way it can affect the prokaryotic cell and not the eukaryotic cell. It's a way of having the medication harm the bad guy and leave the host alone in there. So if we look at a ribosome, they actually are going to have 3 binding sites. So the idea is this first diagram is supposed to be a little bit more realistic picture of what it looks like, being kind of a clumpy protein. If we clean it up, you've got your large and small subunits in there, and then you've got these 3 different sites in there, the P site, A site, and E site. The P site is going to hold the growing polypeptide. Its real name is the peptidyl tRNA binding site. The way I remember it is I look at it and it's going to hold the growing protein in there. And that's, the P goes together with that. You've got the amino acyl tRNA binding site. This is going to hold or accept the tRNA that's carrying the next amino acid. So the way I remember it is, it's in the front, it's the accepting site that's going to take things in. You need up in the roof? The stick, I believe, is in that door, and if it's locked, I've got a key. And then the E site is the exit site. That's where you're going to have the discharged tRNA exit through there. So they come in carrying the new protein, or the new amino acid, it hooks on to that polypeptide that's growing here, and then when you're done with it, you chuck the tRNA out the back end so that it can be recycled. So this shows the process kind of coming through, where you bring this new one in, and now you're adding the piece on, and the other one slides out the back end and is ejected in there. So your translation initiation complex is the union of the mRNA, your initiator tRNA, and the small ribosomal subunit, and then the big ribosomal subunit comes in and closes off the thing and completes the starting unit in there. You've got initiation factors. These are proteins that are required to bring the translation initiation complex in there. And basically, this is a way of adding more regulation into this process in there. They're always going to be synthesized from the N-terminus to the C-terminus. So you've got the amino end, which is your N-terminus, the carboxyl end, which is your C-terminus in there. And then the elongation factors are going to be proteins involved with adding the amino acid onto the previous one. So that is going to be in this area where you're going to start to glue those amino acids together in there. And then the release factor is going to be a protein at the end that's going to help stop binding the codon in the A site. And with the addition of water in there, it's going to hydrolyze the bond so that the completed tRNA is able to exit out the back end. So this is again showing it again. This release factor comes in and it's going to help get this moving to chuck it out the back end. The ribosome separates. Now you've got this free mRNA floating around again. You've got the free polypeptide that's going to go off and do its job in there. So we can have polyribosomes where we will have more than one ribosome read an mRNA at a time. So just like you can make multiple sets of the instructions, you can have multiple workers reading the instructions at the same time. So this one in the first front of the line, you can see its polypeptide is the longest because it's read and done the most of the instructions. The one that's right behind it, it's a little bit shorter. The one behind that's a little bit shorter. The one behind that is yet a little bit shorter. So it works like an assembly line. And each station in the assembly line, you get a little bit more done till you get to the end and you go off and do your next job. And then this is just a picture of it actually occurring in a cell in there. Most of the time these pictures are kind of vague looking because proteins are just clumps that you see. So after we have completed making that target functional protein in there, the polypeptide is going to begin to coil and fold up spontaneously based on the amino acid sequence. So the amino acid sequence is your primary structure of your protein. They're going to automatically start to fold into their specific shapes of their secondary and tertiary structures. A lot of times you're going to have chaperone proteins that are going to help with that. So we have post-translational modification that's going to occur. So after you've made the protein, a lot of times it needs to be modified a little bit before it's ready to go do its job in there. And that's what's going to happen afterwards. So your signal polypeptides are going to be polypeptides of proteins destined for the endomembrane system. In there, it's going to send it off to the endomembrane system so that it can be modified and be sent to do its job that way. Your signal peptides, your SRP, your signal recognizing proteins in there are going to function like escorts to bring the ribosome to the receptor protein in the endoplasmic reticulum. So when it goes into the endomembrane system here, you've got these proteins that are going to guide it in there and help get it straightened out in where it's supposed to go. So it's not just random. So the mutations Mutations are going to be anytime you cause a change to the genetic information of a cell or a virus. Part of it is it does help to create the diversity in the genes, but sometimes it causes changes that we don't really like. So point mutations are changes in just a single nucleotide with its partner in the DNA. So if we look at this little strand here, let's say I have a mutation that occurs right here. This is now mutated to a G. That's an example of a point mutation. So most of the time you're going to have a nucleotide pair be substituted in there. Silent mutation is usually the best outcome you can get in here. When you do a silent mutation, because of the duplication in the code, you change the genotype but not the phenotype. You change what the genetic structure is in there. So it's going to have, maybe say we did CCC here. It still codes for the same amino acid, it just does it a different way. So what you're going to see in the cell and that protein that's made is exactly the same. It just is coded slightly differently. A missense mutation is going to be one that is going to code for something different. So say we changed it to GCG in there. Now you stick in a different amino acid. It's going to make an entirely different protein in there. So if these are so specific that you can take your thread and wind it up in a way, tying knots in it to make a rope this big and have it be exactly the same every time, if you throw off one amino acid in there, you make something that's totally different. And then the nonsense mutation is going to cause translation to be terminated early. It's just going to put a stop to it early in there. A lot of times it just converts something to a stop codon. So the mutation can occur by insertion. So you can switch out something. You can insert something extra. You can delete something. And when you do that, what you do is you change the frame of reading in there. So let's say here, instead of doing this, what we did is we inserted a G in here. So now we don't even have an AUG to get started. But if we inserted another G here, we get started, but our frame of reference that we read everything in is different. So it's changed just about every amino acid in that protein. So this is how mutations can bring so many different variations into nature for things to potentially evolve in there. So this diagram is showing in here how we can have some of these mutations come in here where if something is missing in there, where you get the frame shift, the missense, the nonsense in there. It shows it on a more molecular level in this picture if you wanted to look at it in that frame of reference in there. So what is going to cause these mutations in there? Mutagens are physical and chemical agents that interact with DNA and cause mutations in there. A spontaneous mutation is going to be one that is a mismatch during replication. So it's estimated 1 in 10 to the 10th nucleotides is altered, and that change is passed on to the next generation in there. So you can have them just occur spontaneously as a matter of copying, or you can have something from the outside environment do it. Carcinogens are cancer-causing substances. Most of them are mutagens, so you can be a mutagen and not necessarily be a carcinogen, but cancer is the result of multiple mutations in there. So exposure to a mutagen is never a good thing for a living organism in there. It might be good for evolution overall in the whole population because it will introduce more possible options in, but a lot get killed in the meantime. For the individual, you want to avoid the mutagen in there. And so basically this whole idea of the gene concept is universal, is all that section is on. I wanted to make sure that we got to talk about the operons a little bit in here. The operon is a way of controlling gene expression that has been studied in bacteria. Basically, a cell doesn't want to make something that it can get from the environment. That's a waste of energy in there. And so the operon is going to be a way that it can regulate what's going on, and it can shut off a single gene or a group of genes so that you're not making something you're able to get easily from the environment. So you have a segment in here that is of your gene. You've got the operator that's going to send the DNA. It's a segment of DNA that's going to act like a switch. It may be positioned within the promoter or between the promoter and the genes. But what it does is it's going to control access of the RNA polymerase to the genes. It's going to prevent it from being transcribed in there. The operon is the operator, promoter, and the genes they control. It's the whole unit. You've got a repressor that's going to be a protein that binds the operator, and it blocks the attachment of the RNA polymerase and prevents transcription. And then there is a regulatory gene that's going to create the repressor in there. Sometimes it's located some distance away from the operon, other times it's close. A lot of times it's going to be expressed at a continuously low rate. In the example we're going to look at, the tryptophan operon. And it's an allosteric protein that it's going to have active and inactive shapes. And then a co-repressor is a molecule that's going to cooperate with it and change its shape in there. And being able to watch these in a video is going to make a lot more sense of it than looking at a picture in there. Okay, so I'm going to put it back to the beginning so I can explain what is going on in here. No, it won't get any louder. That's why I get to explain it to you. So you've got your control sites in there, your RNA polymerase that's going to attach on there. Okay. Stop and go back to the beginning. So the trip operon is going to regulate the synthesis of the amino acid tryptophan. And it's got 5 genes in there. Oh my God, would you please stop doing this? So you've got structural genes. And you're going to have so you can see where the tryptophan operon is in there in the circular chromosome, and it's a ways away from it. Okay, so that's a lot nicer when you can actually hear it all the way. Yeah. Yes, inside the bacteria. Yeah, so basically what the tryptophan operon does is it's going to, when you have tryptophan present from the environment, it's going to bind to that repressor protein and activate the repressor so that it can shut down transcription of it, and it doesn't make it when it doesn't need it. And so that the video shows you all of the little regulatory steps, and the link is in here. It's to the video directly, so you can access it off of the Word document, and you can be able to hear it at full normal volume on your computer, which that is the full volume on here. And I either record or it won't, for some reason it will not hook up to the other speakers in here, and we won't get into that. The other type of operon in there is going to be your lac operon is the other one, and there is also a video for that one. And what happens with that one, as lactose is, you don't want to make enzymes for processing lactose when you don't need it. So as lactose becomes more available in the environment, it's going to induce that to turn it on when that molecule is present in there, so that you make the enzymes when you need them to digest it in there. So those are two ways that these are really classic examples, the lac operon and the tryptophan operon, of how bacteria will regulate things. So we're going to stop with that today.